All right. Well, hello everybody, and welcome to today's FlowJo webinar, where we'll be talking about data analysis off of the FACSDiscover platform in FlowJo software. My name is Veronica Obregon-Perko, and I am a scientific advisor for informatics with BD Biosciences, so primarily offering support for FlowJo software as well as general consultation for data analysis. I'm happy you're here today to talk about this more advanced kind of niche topic, but still relevant as the FACSDiscover platform becomes more and more common in the flow cytometry community. So happy to have you here today. I always think it's fun to get a sense of where everybody's joining us from around the world. I know this is a global webinar series here in Atlanta, Georgia, where I'm based. It is about 7 o'clock in the evening. It is a very dark and rainy evening, but I'll won't let that bring down the energy level. Yeah, so go ahead and share where you're from. Always curious to see. Looks like we've got a couple of US-based attendees as well as some people over across the pond in kind of the UK area. So wherever you are joining from, whether it's midday or very late in the evening, very happy to have you here today. So another poll question just to kind of kick things off, given that the FACSDiscover platform is relatively new, I'm really just curious, what are people's kind of learning objectives today, right? What would you say is your experience level with the FACSDiscover platform, and more specifically about the data handling in FlowJo software? So if you all want to just take a moment to share your experiences, have you acquired data on the platform before? But maybe you're just here to learn a bit more about the images, the image handling. Have you acquired data on the platform, but not so much the imaging, but the spectral parameters themselves have seemed a little unfamiliar, so you're trying to learn more about that. Maybe you don't have these cytometers yet, but you're kind of just wanting to learn more about it. Or you have access to them, they're available through a collaborator, through a core facility, so you're just kind of trying to learn a little bit more. So I'll give people a moment just to answer, but it looks like right now we're relatively split between some confusion in handling the imaging component, as well as some confusion in just dealing with the parameters themselves. So I'll give people one more minute to kind of chime in. But the good news is we are going to talk about both of these topics. So in previous FACSDiscover webinars geared in FlowJo software, there was a little bit more of an emphasis on handling the images. And so we will still, of course, talk about that. But I do still think it's worth having a conversation about how the parameters might be a little bit different than we're used to seeing in FlowJo software based on data from like other conventional or spectral cytometers. So there's a couple of nuances for dealing with the data in FlowJo 10, so I'm hoping to kind of alleviate some of that confusion today. Okay, awesome. So thank you for those of you who participated. So let's go ahead and exit out of this poll and get started with our webinar today. All right. Oh, whoops. Meant to just try to get a laser pointer here. Great. Okay, so as I mentioned, right, today's session is going to be dedicated towards data analysis of data coming off the FACSDiscover platform. We are going to give most of our attention to FlowJo software version 10, just because that is, our legacy system, and that is where going to be the most feature-rich experience for dealing with both the parameters and the images. But I will make some mention of version 11 and how it kind of fits into the kind of roadmap for supporting FACSDiscover data. So here's my email following today's session. If you have any questions, don't hesitate to reach out. We're always happy to help. This session is being recorded. The recording will be posted to our website probably within the next couple of days. So you will have an opportunity to revisit the content. If at any moment, if you have a question, you don't have to wait till the end. Feel free to just put your question in the chat. I'll do my best to keep an eye on it and try to, answer questions as they become relevant in the moment. But you don't have to hold your questions. You can go ahead and ask them as they come up. So I'm going to spend a little bit of time in the slide deck today, just kind of introducing, the outline of how our hour together will go. And then I'll go ahead and do the rest of the demonstration in the software itself. I think some of these concepts are easier to grasp when we can kind of see it in action versus, walking through some slides in a slide deck. So we'll start with the slide deck just to kind of introduce some things, and then we'll move on to the software. Okay. So let's go ahead and get started. So here's kind of the learning objectives for today, right? What I'm hoping to cover. So I'll give you just a very brief introduction to the BD FACSDiscover family and the CellView image technology. This by no means will be an exhaustive review of the instrument's capabilities in terms of, the hardware and the resolution during the unmixing process. We do have webinars on the BD website that go into these in a bit more depth, but I think it's worth having a conversation about what the platforms are capable of and how the CellView image technology works kind of at a high level, just so we understand how the images are different from other imaging cytometers. We'll talk about the image features, right? How those reveal distinct information, how we can leverage those in an analysis. We'll talk about data handling. I think that can be a bit of a confusing topic in terms of which files you have to load and when, depending what part of the analysis you're in or what you're trying to accomplish. And then we'll spend some time in the software talking about, prepping our workspaces to, enhance that experience, add a little bit of automation, clean up visuals and views for the data so we don't have long lists of information we don't need. And then, of course, we'll spend some time in the CellView Lens plugin itself, right? How we can view images in conjunction with biaxial plots. And I don't think we'll have a lot of time to go into this, but I do want to at least mention some of our computational solutions for population identification, including machine learning. And then lastly, also if time permits, I'll show you just a quick peek on how FlowJo 11 is able to handle imaging off the Discover platform as well. So let's just quickly talk about the FACSDiscover family and also the CellView image technology. Right, so the BD FACSDiscover A8 cell analyzer and the BD FACSDiscover S8 cell sorter are basically the world's first spectral flow cytometers with real-time imaging. So these allow you to, basically pair traditional spectral data, right, cytometric intensity data that we're used to seeing, like surface CD3 expression, surface CD4 expression, and quantifying the frequency of those parameters. But we also get the imaging component. So at the same time, we can get these reconstructed images and have that quantitative measurement of those image features, which we'll talk more about in a moment. So that definitely sets our platforms apart. So we do have the S8 cell sorter, which has sorting capabilities. This was the first platform of this series to become customer facing. But since then, we have released the analyzer component, which basically has a lot of the same capabilities, but it's a bit more high throughput because you can have a dual mode, with or without the imaging. And it also has a plate loader, so that way you can, capture more samples with a level of automation versus, putting multiple tubes on the SIP if you don't have any interest in downstream sorting. But all in all, they both have spectral effects technology, which I'm not really going to talk about today, but it's just to say that it's an onboard kind of system aware unmixing that helps improve resolution and gives us, better dye options, better opportunities to resolve dyes, and overall just better quality data. So they go hand in hand, their configurations are matched, so it's easier than ever to, analyze a cell type based on its fluorescent and image properties. And then, with the same detector setup and detector array, you can develop a gating strategy to then sort that population should you become interested in functional studies or some kind of sequencing application. So again, just to kind of put it all into perspective of, what this platform is capable of, of course, we have the real-time image cytometry component. That's, of course, the biggest, selling point. But we can also, when you have the S8 platform, you are also given the ability to sort a population. So with the A8 analyzer alone, right, we can analyze those cells, but we can't separate them from other populations, right? When you have the S8, you get that capability as well. And we can help you develop computational sorting strategies with Hyperfinder. This is a tool available in FlowJo, which we'll talk about at the end if we have some time, but essentially it gives us an opportunity to define an otherwise very complex cell type to identify. We are given a gating strategy that we can then directly import inside Chorus and use it to sort that cell that we've identified. 78 detectors, so very powerful spectral cytometer, lots of opportunity for various dye choices. And then, of course, we have the imaging detectors off of the blue laser. So 3 imaging detectors, which can be used for the imaging component. And then, of course, the remainder 70 plus detectors, which we've previously published. We can do up to 50 different colors on there. Okay. And then, of course, we can have plate-based platforms, 6-way sorting. This all has more to do with the handling of the samples themselves. But just to give you an idea that, it's a very powerful instrument. We can get more measurements than we ever have before. Okay. So what's distinct about the CellView image technology itself, right? So it's important to note that the images that you will see off of these platforms are not based off a shutter system, right? It's not like the cell passes through an interrogation point and pauses for a brief moment so that we can capture like an actual photograph. But instead, these are actually reconstructed images, and the images are reconstructed based on these waveforms, right? So there's actually a video here that I think describes it best. But essentially, instead of having one single beam, that beam is divided into hundreds of basically beamlets, right? And the cell passes through these beamlets, right? There is a pulse or a signal that's collected for each of those beamlet regions, right? So those pulses are then gathered and reconstructed into this image that you see here, right? So it's going to look different than what we're used to seeing, say, for microscopy or other image-based cytometers. But because of that, we're also able to collect more physical information about the cells in the form of actual measurements, right? Not just the image itself. So what are those imaging measurements that we can obtain from this platform? So it's not just, again, just the pictures, but also parameters that we can ultimately gate on. Okay, so these are all features that come off of the imaging detectors. I'll break them down very briefly here, but just to show you, the application that they can have, the utility that they can demonstrate when paired with a typical cytometric analysis. So for instance, we can have something like eccentricity, which can reveal information about cellular morphology. And so if we have a low eccentricity, this could mean that the signal itself is kind of round and rotund, as where something has a high eccentricity, it's a lot more elongated, kind of cigar-shaped. So that can apply to not only the target signal, like some kind of a dye or marker, but even the shape of the cell itself, even if we don't have any dyes in place. And so this histogram is just to show you that, this parameter can basically be plotted on either a biaxial plot or a histogram, and we can gate and subset populations based on quantitative measures of these image features. So it's not just staring at the pictures themselves, but we can also draw gates to get closer to a cell type that is very elongated or very rotund, right, very round. Right, there are also things like max intensity and size, which, together and separately can reveal information about the brightest part of the signal or the total brightness of the signal. So they can be correlated. But you can see here again, if we gate on things that are high for max intensity or high for the size, right, these are going to be very bright signals. Versus if we do something that's low max intensity, low size, these are going to be either dim signals or maybe a diffuse signal, but it's still quite dim. Yeah. So again, these could also have some application for tracking something intracellularly or looking at different cellular morphologies. Radial moment is an interesting one as well. So this is going to reveal, whether a signal is rather like a punctate or whether it's more diffuse. And so I see this used most often for looking at nuclear translocation, for instance, right? Rather than having something diffuse in the cytoplasm or expressed on the surface, we're looking for more of a distinct punctate signal inside of the nucleus. So something with low radial moment would kind of fit into that category. Correlation is basically the coincidence of any two channels. So if we mark something as red and green, right, when they're coincident, they will fluoresce yellow, or I should say they will have kind of the visual profile of being yellow. And so again, this could also have utility for nuclear translocation, right? If we have some kind of a marker that we're looking for in the nucleus, right? I could maybe look for it to co-localize with DNA content, a DNA content dye. And if I see yellow, then I know that the green and the red are coincident. And so I have more evidence that those two are kind of hanging out in the same place in the cell. And so that can be a very powerful parameter as well, or looking for two interacting proteins. Delta center of mass, very similar, right? The distance between two signals. Diffusivity reminds me on some level of radial moment, but rather than a complete circular radius, right? Again, more of a diffuse signal. So something that would be possibly in the cytoplasm versus something that also represents like a punctate signal. And so you can see how there might be some kind of overlapping physical features that could be identified with multiple of these parameters, right? So if you were looking for some kind of a signal trafficked to the nucleus, right, something like diffusivity and radial moment together might help you get closer to that phenotype, right, by gating on these properties. Okay. Okay, so I wanted to spend a few minutes just talking about data handling, particularly for the image analysis. Okay, so when you acquire your data off of the FACSDiscover platform, right, this data is going to be acquired and exported from BD FACSDiscover. Okay, this is the software that operates the cytometer. At the end of your experiment, you will of course have an FCS file that is standard for any typical flow cytometer. This is where your individual event information will be and have the cytometric intensity data. Now the images will be placed into what's called a waveform file. It's a single file where your images are kind of all zipped together. But this file isn't directly readable in FlowJo software. It is required that it be extracted for all of the TIFFs. So each of your cell-associated images is represented by a TIFF file, but those TIFF files are zipped into a CVW file after exporting from the cytometer. So before you can load your FCS file and see the corresponding cell-associated images, the TIFFs have to be extracted from the CVW. Okay, so what are your options for extraction? There are two. One is you could use the BD CellView image extractor. This is a program that is typically found on the computer that operates the cytometer or the sorter. Okay, so it will be an onboard program. You just run the CVW file through there and it will provide the TIFFs, which you can then relocate to a server location or something like that. Now, if you would prefer to just migrate a single CVW file to some kind of storage location, you can also do the image extraction in FlowJo software version 10 using the CellView Lens plugin. Our newest plugin, 2.0.15, does have TIFF extraction capabilities. Okay, so if you have an older version, it's not capable of doing that, but this newest one does. So if you install that new version, you can load your CVW. A dialog will come up when you launch the plugin from the FCS file in the FlowJo software, and it will just ask you, where the CVW file is stored, and then it will, do the TIFF extraction and keep those files in a folder nearby. Okay. So long story short, those TIFFs have to be extracted from the CVW, either from the CellView image extractor or using the CellView Lens plugin. Okay. So once you have the TIFFs extracted, really all you load into FlowJo software is the FCS file, right? We do not load the TIFF images or the CVW directly. It is just the FCS file, but when you launch the CellView Lens plugin off the FCS file, it maintains the connection to the extracted TIFFs. And so throughout your analysis, right, you might become interested in a population that you want to computationally define. You could, of course, manually define it, but if you decide you would like to let an algorithm find the best gating strategy to find that cell type again, once you have those algorithm defined gates or gates you define manually, you can then import your workspace directly back into Chorus. Okay, so it is a round trip where you can leave Chorus with your data, analyze it in FlowJo software, and after you've obtained some kind of gating strategy that you then want to see in Chorus, you can do that directly with a workspace. Right. So when you have your file stored locally, right, it's just a matter again of loading the FCS file. Once you have the CellView Lens plugin installed, which is available from the FlowJo Exchange, and just needs to be installed into your plugins folder. Once that plugin is launched off that FCS file, it's going to just ask you to point to where the images are. It can typically find it automatically as long as you have the images stored right beside the FCS file, and you follow this precise folder structure, right? After your images have been extracted, they typically will come in these folders with these numerical strings. So avoid the temptation to rename the folders, relocate them, move images around, because that folder structure is very sensitive to breaking the communication between the files, okay? So whether you extract from the extractor program, the image extractor program on the Chorus instrument, or whether you use the plugin, the folder structure will be basically the same. So just avoid messing with that or renaming it, and it should be fine. And once that connection is established, then you're ready to continue with your analysis, right? So you can launch the window and you'll be able to interact both with the FCS event information and the TIFF images. Now, another option, if you prefer to kind of keep data on the cloud storage or something like that, or for whatever reason cannot use the plugin and didn't have an opportunity to extract the TIFFs at the cytometer, we do have TIFF extraction capabilities on the BD Research Cloud. The BD Research Cloud is a ecosystem for supporting flow cytometry studies. It has a lot of panel design capabilities, but it also has a data storage component. With a free account, you can store up to 1 GB of data. You can also make a collaborative organization where you invite users, and for every user that's invited to the organization, you get an extra couple of gigs of data. So worthwhile to make a free essentials organization with a handful of lab mates, because you could collectively get up to 50 GB of storage. So depending how much storage you have, right, that might be enough to upload both your FCS file and your CBW file. And then what will happen is when you're in the BD Research Cloud, uploading your CBW file, it will ask if you would like to extract the TIFFs kind of in the background and keep them stored on the cloud. And if you enable that, then you basically have an opportunity to add your samples from the BD Research Cloud. And when you launch the plugin off your FCS file that you brought in directly from the Research Cloud, it will automatically have that connection to the extracted TIFFs on the Research Cloud. Okay, so you don't have to store the images locally. You don't have to open up this dialog to connect the images. That this smart connection is maintained. Okay. Now the caveat here, I mean, with any cloud storage is it's of course sensitive to internet connection. So you'll of course need to be on the internet to download, upload, at least initially. So that's gonna be dependent on that. And when you have data stored locally on your device, it tends to just load faster, just because it's already nearby. So if you have a really, really large data set, you might experience some lagging, and the seamlessness of the analysis might seem a little slow. So I think it's okay for small to moderate sized data sets, but you might see some lagging for larger data sets. But it is an option. It helps keep the data mobile and it makes it easy to share the data. So just wanted to point that out as an option. Okay. All right, so the rest of the time, I'd really like to talk about just getting started loading your data in the software, right? How can we improve the experience? What are some of the kind of pre-processing steps we can take? And then ultimately, how do we interact with the CellView Lens plugin? Okay. So I think at this point, I'm going to go ahead and move into the software. That way we can actually see where this functionality is and kind of learn where to access it as we start to deal with our own data. So I'm going to exit out of the slide deck here. Whoops. There we go. And move into our demo. So I'm also going to turn off my camera just to give myself more room here on the screen. Okay. Okay, so one thing I want to point out before we start getting into the weeds of the workspace is I just want to take a moment one more time to talk about folder structure and files. Okay, so notice how I've only loaded an FCS file here into the workspace, right? We do not load the TIFF files or the CVW files, okay? So again, folder structure is important, okay? So notice here, this is the experiment that's been exported off the cytometer. Okay, so I have the FCS file, which has kind of the cytometric information, and the CVW has been extracted into these two subfolders, each containing, hundreds possibly of different TIFF files. Okay, those TIFF files will be stored into kind of these subfolders. You may only have one, but if that folder gets full, it moves on to the next and so on and so forth. So you could have one or more of these subfolders, but keep those subfolders in the parent folder. Don't move any of this. Don't rename it. Same for the FCS file. Keep it nearby. And before you extract the CVW file, also helpful for both the CVW and the FCS to have the same name. Okay, so again, avoid the temptation to rename things and move things around. So just try to keep that preserved. Okay, so if you are going to relocate data, try to move it as a unit rather than moving only components of it. Okay. Okay, so at this point I have loaded, or I've preloaded, I should say, an FCS file here. Okay, so this is some data off of the S8 platform. So let's talk about these parameters. So if I launch a graph window here off of that FCS file, for those of you familiar with FlowJo 10, you know that we have a parameter drop down for gating purposes. So X parameter drop down, Y parameter drop down. And so when looking through this parameter list, you'll of course see the things you're used to seeing as a cytometrist. You'll see the scatter properties. Your raw detector information will be here, right, unlabeled. So it will say things like V1, V2, R1, R2. And then you'll have your unmixed parameters as well, right? So those are going to be here. I can tell because they're labeled. So here's my CD1C BV421, CD3 BV395, CD14 BV786. So these are my unmixed parameters that I'm most likely going to leverage during gating and population identification. Okay? But with the Discover platform, you are also going to get a ton of extra parameters you're probably not used to seeing. A lot of those will be image-derived features, right? We talked about these in the presentation. So things like max intensity, center of mass, radial moment, eccentricity. So that's a lot of image-derived features there. You're also going to get all of these extra parameters for the raw detectors, right? In addition to area and height, you're also going to have width, and you'll also have something for the T as well, right? Which I think is for time of flight or something like that. So needless to say, this is a really long list of parameters. Now I could utilize the search if there was ever anything I specifically wanted, but for those of us that are kind of used to browsing through scrolling, it's going to be a little cumbersome. Okay? So one way we can kind of filter down that list is in our preferences. So your preferences is this heart-shaped symbol here at the top of the workspace, right? When you hit that button, it's going to open up this preferences window. In the cytometer section, you'll have an opportunity to set some kind of default rules for dealing with these parameters, okay? So here you can see I have the FACS S8Discover, right? This is the cytometer that this data was generated off of. You'll see that cytometer listed here when you load data from that cytometer. Okay, so if you've never loaded S8 data, into FlowJo, you won't see the S8 cytometer here. Okay, so it does require that the data be loaded at least once for it to show on this list. But once it shows on this list, right, if we give our attention here at the bottom, right, there's an opportunity to kind of add some filters so that we're not always having to scroll through that long list. Okay, so this is kind of the recommendation for dealing with S8 or A8 data in FlowJo 10, right? You can create a rule to hide -H parameters, hide -W, hide -T, right? And you can do this by adding a rule. So here, I'll go ahead and delete these so I can show you. So if I want to add a rule, I'll say, okay, well, I want to hide anything that has a -H. And also if it has a -W, okay? And also if it has a -T, right? So if it has this or this or this, I want them filtered out of that list, okay? Now this part's optional, but let's say you like seeing the scatter property for these, okay? You could create a rule that says, well, if it has these, but not, right, something like SCH, right? So I still want to keep the height parameter for my scatter or something like that, okay? So you can create these rules and that'll help filter that list by like 70%, okay? And you only have to do it once. Now, keep in mind, anything you change in your preferences, you typically won't see the impact until you do a new workspace. So I'd recommend making this a part of your initial workflow as you onboard data from that cytometer. Okay, so it's worth loading in some data and setting these filters because they'll only apply to subsequent workspaces, okay? So that's one thing about those parameters. So now another thing about the parameters that we can talk about is scaling, right? So that's going to be another thing that might seem different than what you're used to, right? So I'm going to go ahead and just leave this plot here. But let's say, for instance, if I look at something like a BV510, right, you can see that this is a, fairly bright dye, right, going all the way to 10 to the fifth. But the range on the S8 and A8 is actually quite large, right? I believe it can go all the way to like 10 to the seventh or 10 to the eighth. So it is possible you could have dyes up in that area. Okay. The other kind of parameter that's not super intuitive to set are the imaging features, right? So as we start navigating some of these image-derived features, right, you can see the scaling is a bit inadequate, right? Some of the parameters only have a scale of like 0 to 1. Some of them will have a scale of like 0 to 10,000, right? Some of them will be better linear, some of them will be better log. And so, we're kind of learning as we get more exposed to this data, but typically the default scaling is just not going to handle it very well. But the good news is we've been learning a lot from this data so we could start giving recommendations for the scaling. And we've compiled all of that scaling recommendation into some documentation on our website. So if you go to flowjo.com and go to our version 10 documentation, we have a section on advanced features. Right? When you click on the advanced features, we have a section for S8A8 data analysis, and in there a subsection for FACSDiscover S8 scaling. Okay, so even if you can't find this page again, typing it into Google will likely result in it. And so on this page, if you scroll to the bottom, you can see that we've given kind of a cheat sheet, right? What we recommend you use for scaling that data. Okay, so you can see that there's scaling even for the forward scatter, and you can see how quite a big a dynamic range it has. We also have recommendations for the radial moment. Notice how that one looks good in log, but eccentricity, which is just above it, right, it basically goes on a scale from 0 to 1. Okay, so we've kind of just learned from the data and given you these recommendations so that you're not always having to adjust these in the transform property. Now, of course, if you're an avid FlowJo user, you're very used to having a graph window, hitting this T button, going to customize axis, and, doing kind of these one-off changes. And that's not to say you still can't and that it won't ever be necessary, but you'll find yourself doing it a lot the more and more you work with this data. Okay, so it's helpful to have kind of some default rules. So how do we make those default rules? Well, that'll be back here in the preferences. Okay, so if you hit that preferences button, we go into cytometers. Okay, here there's an opportunity again to go back to that Discover platform. Here at the bottom, advanced rules. Okay, so when you click that button, this is where we can start creating rules for this platform. So you can see it's even reminding me which cytometer I'm using. So if I want to add a rule, I can create a rule. So let's say this one, I'll call it eccentricity, perhaps. So letting me rename it. Come on. Huh. Not sure why it's not letting me rename it for some reason. Maybe it needs to add a condition first. Okay. So let's see. So then you can go ahead and add a condition. So there's various kind of Boolean logics here. So you could say something like what kind of a rule are you looking for? And let's see, maybe I'll say something like parameter name contains. Okay, so I'll say okay. And so here you're going to see a list of parameters, and it's basically telling you which parameters are going to be impacted by the rule that you make. Okay, so maybe I'll say something like if the parameter name contains eccentricity, make sure I spell that correctly. Right, you can see here it's telling me, well, these are the parameters that will be impacted by the rule. Okay, so if the parameter name contains eccentricity, right, I can scroll through the list and make sure there aren't any others that I wanted to be affected by this rule. I can come into then the transformation settings and set the rules that were in this cheat sheet. So here it's recommending I do 0 to 1 on a linear scale. Okay, so I'll just go ahead and do that. So I'm going to change this to linear, right? I'll make the minimum 0 and I'll make the maximum 1. Okay. Right, and I can see in this data, I actually have some stuff kind of going off scale. So maybe I'd want to add a little more than 1 something if I wanted to. Okay. Right, and so once that condition is done, you can add multiple for the same parameter. Like I could say, contains this but doesn't contain that, and that way if I had specific eccentricity parameters. So you can get incredibly granular with this. Now it's letting me rename it. I guess I had to add a rule first. Okay, so there's my rule. Okay, and so you can come back and edit these, as necessary. Okay, and then I'll say okay. So you can see there it's telling me how many rules I have applied to this data set. Now again, keep in mind this will only apply to new workspaces. So again, it's worth kind of doing some homework beforehand before you get too invested in your data because you'll have to do a new workspace to see the changes. Okay. All right. So you'll notice my list won't be filtered because I'm still in the same workspace. But one thing I wanted to show you while we're still on the topic of parameters is that we talked about the scatter, right? The image derived. Here's your unmixed, which are labeled, right? Here's the raw detectors, which are not labeled. They just have the detector name. So some of you may have noticed that when you load your data, if you go to the very bottom of the list, you might notice these parameters here, right? Notice how they're labeled with my target and my dye, right? CD1C, BV421, CD45, BUV805. But they have this SW unmix in the name. And they also have a detector that I know for sure was not the primary detector I assigned it to at the cytometer. So these parameters are actually just kind of an artifact of FlowJo trying to assign a primary detector to the parameters, right? In the chorus files as they stand, there's no information on there that allows us to be able to assign a parameter to a primary detector, or at least know what the user assigned that parameter to as a primary detector when they were at the machine. So when FlowJo detects the acquisition defined matrix, it's just trying to assign that parameter to a primary detector. And if it doesn't have that information, it's kind of just going in order of the detector array. So, right, so it starts with UV1, goes all the way to 11 in this case. So it's, nonsensical, really, but it's not causing any harm, right? So for instance, if I just duplicate this plot and let's look at something like CD1C BV421, and then I'm going to look for that same SW counterpart. So here's CD1C BV421 SW1 mix, right? Those data distributions are the same, right? So whether you initiate your gating on this one or this one, it doesn't really matter. The biology is the same. So you can basically ignore the SW ones and give your attention to the others. Now the caution will be if you decide to edit the unmixing matrix, that will only impact the SW parameters. Okay, so if you edit the matrix, now you have to give your attention to the SW. If you decide to recalculate the compensation, that will write new comp parameters, and now you can't use either of these, right? You'll have a new set of compensated parameters, okay? So long story short, it doesn't matter which one you use on the data if it's completely unchanged. As soon as you start tinkering with the compensation, editing the matrix will only impact the SW because it's the only one that received the application of the matrix. And then if you calculate a new matrix, you'll get new parameters that say comp. Okay. So just keep that in mind. And even though your unmixed parameters that come written into the file don't have that matrix is applied, right? So this data is unmixed. And if you did do spectral effects at the cytometer, that is also applied, right? We don't really have an indication or a symbol to tell you that, but it is there. Okay, so you don't have to worry about reapplying it or anything like that. Okay. Now, just to say, if you calculated unmixing at the cytometer, there's generally no need to redo it or edit it. If everything went well at the machine. If you want to peek at your matrix, I mean, you can do that. So you'll have this matrix icon beside the unmixed file, right? When you double click it, you're going to be brought to the matrix preview, okay? But this is a little hard to interpret at times because of these UV detector assignments that we talked about. Okay, so if you see this, don't panic that we have not redone the math. We haven't, somehow thought that your single stain controls were acquired on UV detectors or anything like that. This is still your spillover values as they were calculated at the cytometer. But again, because normally in a matrix we're looking at the detector information, FlowJo just thinks these were the detectors that were used as the primary when we were looking at your single stain controls. Okay, so the spillover values are still correct, but it can of course be a little tricky to understand what you're looking at, right? You could look at the n by n plot, and that might be a little bit more helpful. But this also makes it tricky to edit the matrix. Okay, so again, not something we recommend doing anyway. But if you did want to do it here, it does make it tricky, and also sometimes users experience some lagging, some crashing if they have a lot of single stain controls. So my advice would be if you're unhappy with the compensation or a mixing, it's probably going to be easier to recalculate than to try to tinker with the matrix. Okay, it's not very intuitive. So if you want to recalculate compensation, we have webinars on that, so I won't dwell on it too much. I'll just point out the S8 specific things. First thing being that you of course have to have your single stain controls. They have to be in the compensation group. That's all true. But the thing to point out is that when you're in the compensation wizard, okay, so when you're assigning your single stain controls to the detectors, make sure you have the spectral checkbox enabled. In the All Detectors button, make sure you turn off the imaging parameters, right? Because we're not using those in the unmixing process. We just want to use the raw fluorescent detectors. Okay, so make sure you deselect the imaging detectors, deselect like the T, the W, the H, just pick the area for the raw detectors. Okay, don't pick any of the image derived features because those channels are not helpful or those measurements are not helpful for unmixing. Okay. So make sure you do that. And then when you first launch the wizard, you'll get a dialog that looks a lot like this. And make sure you do the same selection, right? In that window, it's going to be asking you to pick the detectors you want to assign single stain controls to. So of course, we don't have single stain controls for the imaging channels, so just deselect that, okay? And then everything else is business as usual, right? Once you assign your single stain controls, to the detectors, right? FlowJo will do that for you based on the brightest detector. It typically gets it right, but you can change the assignments, right? We need an exemplar negative, exemplar positive. You can preview those plots down here, okay? And then you can say view matrix, and you'll basically be brought to a window that looks a lot like this, okay? Right, so I don't want to dwell on that too much because we have webinars on compensation. It's very much the same process, but again, the call outs are just make sure you exclude the spectral or exclude the imaging detectors, right? We just want the fluorescent channels. Okay. Okay, so now, so let's say this data, right, I've already spent some time in the workspace filtering parameters, and I did some scaling. I'm generally okay with the compensation. I don't want to do any new unmixing or anything. So let's talk about the CellView Lens plugins. I'm actually going to open another workspace because this one, although it has images, I don't find them to be super biologically interesting, but I liked this data set because it had single stain controls. So I'm going to just open up a different data set that I think that lends itself better to demo for the imaging properties. Okay. Mhm. Oh, whoops. I guess I already had this open. Here we go. Okay. All right, so this is a fairly simple experiment. It's just a three-color imaging experiment, and we're just looking for GFP expression, right? Just visualizing GFP here. So here's the FCS file. So a reminder, right? You need to have the FCS file loaded into the workspace. And then I'm just going to show you again the folder structure here. And then a reminder, right? I do have the TIFFs extracted, and right, I do have those that subfolder structure there preserved. Okay. Okay, so once you have the FCS file, right, the BD CellView Lens plugin can be retrieved from the FlowJo Exchange. Okay, once you have that installed, it's available here in the plugins, so you can just select the plugins drop down, right, BD CellView Lens, right, and when you launch that, right, it's going to open up this dialog here. And actually, I had one already open before, so I think I'm just going to use that one. Here we go. Okay, right. So here is that data set there. If you're having some trouble connecting the images, right, you can also go into the tools and say open event to image file mapping, right, and try to kind of help manually connect those images. So here it's giving me a green check mark saying it found the images just fine. If you see a red X, it is struggling to find the images, so you'll have to kind of help it along, right? And usually, if you have the TIFFs nearby, if it's having trouble, it's just because something's been disrupted with the folder structure. Okay. Okay, so here's that data loaded in here. Okay, so let's take a tour of what we're seeing in that CellView Lens window. Okay, so here we have kind of the image gallery, the image wall. Then you have a bigger preview image here. Now, one of the first things you may want to do is kind of do some changes to the filters here. Okay, so if you want to change anything about the filter properties, that's this button here, right? It looks like kind of like these filter toggles. Okay, when you hit that, you'll be brought to the image filters area. And so here you have an opportunity to just kind of set some color palettes, right? Help yourself with the interpretation of your data. So what you're seeing here is each of the imaging detectors, right, which you can rename if you would like. And then here you have an opportunity to assign the colors for the different imaging detectors, right? So if I, for instance, want to keep green for the GFP, I can do that. If I have another marker, let's say drag 5 in the third imaging channel, maybe I'd want to make that red or something like that. And then if you have some kind of surface marker, right, maybe I'll make that blue. Okay. And so you can see you're changing the color, right, of each of those different imaging detectors. And over here on the left, right, you have an opportunity to decide what you want to be part of the overlay. Okay, so you have the three imaging detectors, right? The last row here showing you the overlay of what you have selected. But you can also have other features that might be helpful to kind of cast a shadow on the cell because maybe you didn't have a surface marker and so you want to see the boundaries of the cell. It might be helpful to turn on something like the side scatter or the forward scatter or even the light loss, which is like the shadow of the cell. Okay, so I have them off, which is this yellow X, but if you double click it, it kind of enables it back. Okay. So I'm going to leave those off because I don't think that they're needed here. Okay. We have this remove background button, right, which can be helpful if to remove some of there might sometimes be some background noise in some of the channels, right? You can hit that and it'll help remove kind of that background noise from the images if you would like. Okay. Let's see what else here. You can do multiple filters here. So you can see there's different ones, some to help you along. We'll import the one that you established at Chorus, but if you would like to make copies or edits, you can also hit this plus button to create a new image filter. So you don't have to overwrite the changes from Chorus. You can make new ones. You can also create a copy so you don't have to start from scratch. Okay. So a lot of opportunity here to change the visualization of the cells, right? But keep in mind, at no point are you changing kind of the raw information, right? That raw image derived signal for how much, GFP intensity is there. That's not going to change, right? This is just kind of adding like a coloring to that signal just to help with presentation. Okay. So once you're happy with the things here, right, there's also some opportunities to change like the contrast and the brightness, some, kind of things borrowed from the microscopy field. But we have documentation on all of these features, so I'm going to just kind of move on, right? But once I apply those filters to the workspace, right, you can see here that this is what we get, right? So I have the preview there. If there's a cell I want to see in particular from this gallery, it's interactive, so I can just start to, look for cells of interest. Okay. We also have different values here to help you, more so like if you're interested in identifying the center or edges of cells, particularly for like cell-cell interactions. So if I turn on, for instance, like regions, right, that converts this view here almost as if like how many different regions are detected in a single image. Right here, I can detect 3, here there's 1, here there's 2. So if you're trying to eyeball kind of multiple cells in an image, that can help out. Right, we can also outline the edges of the cell. We can have connections between cells. So for instance, here I can visualize this connection between these 3, possibly interacting or at least neighboring cells, and establish a centroid calculation as well. Okay. So those are all just visuals that can take place here within the plugin. All right, let's see what else do we have here. So other features here in this button. So there's also an opportunity to interact with your images and the workspace. Okay, so if we hit this kind of biaxial plot button, right, this unlocks kind of the mouse over seeker function. So you'll see that it's yellow. When it establishes the connection with the images, it'll turn green. Okay, once it turns green, right, you can see that if I move my mouse inside of this 2D plot, I can kind of visualize, right, what cells I'm looking at in that part of the plot. Okay? So let's take a look at kind of a more practical example of that. So let's say I'm interested in wanting to know whether a cell is truly GFP positive or not. Like has the GFP entered the cell, or is it just maybe some background noise or some kind of artifact of the surface staining, right? So now I can take that node and maybe I'll just pop it onto these GFP positive, negative, and unknown gate, right? So let me go ahead and launch it off of something like this. Okay. And actually, I'm going to drag it under that as well. And let's go ahead and give our attention now to that singlets population, right, where I'm trying to interrogate the different GFP signals. So I'll go ahead and hit that 2D plot, right? And so the question here is, I know for sure these are negative, I know for sure these are positive, but I'm less sure about the ones in the middle, right? So if we could use the mouse over function, right now I can see that actually most of those kind of GFP unknowns, right, like I don't really see a ton of green in there. But if I go into the GFP positive gate, right, I start to see more of that green signal. So now I have like visual confirmation that even though this unknown gate, one might argue is kind of GFP intermediate, right, there isn't actually a whole lot of discernible GFP signal in those cells. Okay, so that's how one utility of kind of combining those two visuals. You can also sort within this window. So perhaps I do want to give my attention to the GFP intense ones. I could say, well, I want to sort this view maybe by one of my GFP parameters, right? Maybe I'll say I want to see, let's see, what would be a good one? Maybe like total intensity or size. Right, so if I say size, it's in ascending, so maybe I'll do descending order, right? So now I've sorted this view by I should be having my most intense GFP signals here at the top, and then becomes a little bit less intense as you go along. Okay. Now, image sets can also be a very powerful feature here. So if you start to build curiosity in certain images, right, you can use the show image sets functionality here. and start to curate images of interest. So when you hit that checkbox, we can also unlock that ability to make an image set. And maybe I want to build a set of images that, have really good central GFP expression. So I'll start just clicking images and kind of building my list here. Right? And you can see that I'm kind of making a list of GFP images that I like that I'll want to revisit at some point. Okay? And when I'm satisfied with the list, I can lock it. And I can always come back to this set. But one thing that could be of incredible value is to have these cells kind of grouped together. Because even though these are images of cells, right? So they ultimately represent cells I'm interested in. Well, there's a button over here where I can say create new population. And now I have them here nice and handy, right? All 8 of those events. Of course, this could be a much larger list, but I'm sure you get the idea. So here I can have those 8 images that I can always go back to and the representative cells. Okay, and then there's like a not gate, which is basically everything that was not in those 8 images. Okay. So image sets can be very powerful. There is also a nearest neighbor calculation as well, so a little bit of machine learning. So if you are building a set of images, maybe for a nice nuclear translocated signal, you could tell FlowJo will find me images and cells that look like these, and it'll have kind of the same result, right? You'll have those 2 subpopulations, right? Everything that was off target and on target. Okay. So we have another webinar that's spends like a whole hour on the machine learning capabilities of CellView Lens. So I'm not going to spend time on that tonight because I wanted to spend more time on kind of like the data pre-processing. But I'd invite you to watch one of those. And if you are interested in kind of machine learning, neural network modeling, if you hit this button over here that has kind of like a gear and the brain kind of icon, this is where all of our machine learning capabilities are. So again, it's a bit of a time suck. So I'm not going to spend time on it today, but just know that they're available, right? We can try to help ourselves automate the identification of different cell types with some machine learning. Okay. Okay, so if I leave the image sets here, right, there's a couple shortcuts for you. If there's an image set that you like, you can hit this camera icon. It'll send that over into the layout editor so you can get those all curated together. If you hold the shift key and hit the camera icon, similar view, but this time you get everything that was kind of in the big preview as well as the overlay channels. Okay, so that's one way to kind of snapshot those images for you. Okay, let's see. I think I covered, for the most part, everything in the CellView Lens window. I mean, it can definitely do a lot more, but the goal of today's webinar was to focus on S8 data in general and not just the imaging component. So you're getting kind of just a taste of that. Okay. All right, so I think that's good for the images. So let's see. So we have maybe just a few minutes left. So I'll wrap just to say that we do also have the ability to see images in FlowJo 11. So I'm going to minimize this workbench and open up FlowJo 11. FlowJo 11 is available for anybody who has a FlowJo 10 portal license. Okay, so it is authenticated through the BD Access portal. But it's your same FlowJo 10 portal credentials. Okay, so you'll notice here when I launch the BD Access portal, which is a separate authentication program that gets installed, I'm still just putting in my FlowJo 10 credentials. Okay, so you don't have to make a new BD Access portal account. You don't have to get a new FlowJo 11 license, right? This is all still through FlowJo 10 portal licensing. Okay, I'm going to sign in. Okay. And I'm just going to open up that same data set we've been looking at, but this time we're going to look at it in FlowJo 11. Okay. All right. So for those of you that haven't seen FlowJo 11, we have webinars that spend a whole hour in this topic. But what I really want to just show you is that we can support images here, and we're working to build that functionality, and it keeps growing with every quarter. Okay, so for now, if we go down to instance for my singlets population, right, going back to that same question of negative, positive, and questionable. Right, I could turn this into here where the graph gallery. We have an image wall feature where we're able to retrieve imaging information from the extracted TIFF file, so similar folder structure, but all that same functionality, right? And I can even make this a dark theme, which is always my favorite to do for imaging data. Okay. So I can still, right, select images to preview them. I can change colored overlays, filter information, brightness, contrast, all of that. We do still have kind of the seeker function. So if you come into this little, looks kind of like a target symbol. If you click that, right, you can still get that same mouse over functionality we talked about, right? So a lot of interaction with the images. I can also pick an image and then ask to be shown, right, the corresponding event on the biaxial plot. Okay, so these sections all coordinating with one another. Okay, and the image wall will change depending on what population you have selected. So here's all my GFP negative, no surprise, I don't see any green there. Here's all my GFP positive, right? And I can already appreciate kind of the biological distribution of those signals. Okay, so just to say, we haven't left imaging behind in FlowJo 11. Machine learning is coming, but for now, if you're looking for just visual confirmation of gates, we can already provide that for you. Okay, so I see we're just about out of time, so I'll go ahead and start to wrap up here and see if any questions come up. One thing I'll say as well, I want to unhide this slide to show it to you, is that the BD Research Cloud, right, I did talk about it and its ability to support data handling. But if you have any questions about panel design for the spectral cytometer or dyes that are compatible with the imaging detectors, we do have guided panel design for the S8 and A8 platform on the BD Research Cloud. And it's guided in the sense that we recommend dyes for the imaging detectors, and we also warn against dyes that spill into the imaging detectors because we want to keep those very clean, right? So we get nice, good, resolvable images. So if you're looking for panel design resources for this platform, I highly recommend you check out the BD Research Cloud. And again, in addition to panel design, it also does data storage and sharing. So I'll go ahead and say we are starting something new on the FlowJo team, right? We know that these webinars can be a lot of information, and while it's fresh in your mind, you may have questions that emerge, right after the webinar, or maybe even a week later, and something you wish you had asked during the webinar. So we're going to start doing these kind of virtual office hours. So we'll designate a day or two following the webinar of a specific topic where you can schedule time to meet with us and ask deeper questions about this topic. Maybe you can bring your own data. We can walk through it and talk about any confusion that might still be lingering around the topic. So I'll leave this QR code up for a moment, but essentially I've blocked off the day of October 8th, so a few weeks from now. And so if you would like to book some time with me to talk about your S8A8 data analysis, whether it has to do with the imaging component or just high parameter spectral cytometry, I am happy to support that. So you can just scan that QR code. It should bring you to a Microsoft bookings page, and there you'll have an opportunity to book an appointment on that day. Okay, and if there's anything you want to send beforehand or share beforehand, I'll also go ahead and provide my email address once more. So here's my email address. If you have any questions about the booking link, setting up consultations, additional educational resources, don't hesitate to reach out. And I'll pause and see if there are any questions, though I haven't seen any come through in the chat. But I'll hang around for a minute after stopping the recording. Otherwise, I will wish you all a good rest of your day, and thank you so much for joining us today for this educational series, and I hope to see you at one of our future events as well. Thank you very much.